something hiding inside all of us. Deep within our DNA, we harbor the remnants of ancient viruses. These viruses, known as retroviruses, infected the reproductive cells of our ancestors millions of years ago, and now they are inside all of us as endogenous retroviruses. Today, these endogenous retroviruses make up over 8% of our DNA. One of these endogenous retroviruses is called endogenous retrovirus K, or ERVK, and it has the ability to produce an important protein called integrase. Integrase functions by inserting viral DNA into host DNA, and this is how these viruses infected our DNA millions of years ago. Through this process, integrase causes DNA damage, and integrase itself does not have the ability to repair the damage it causes, and instead, it relies on our host repair mechanisms to recognize the damage and have it fixed. If this DNA damage is repaired successfully, the virus is integrated into our DNA. But this is not always the case. Sometimes that DNA damage is left unchecked and can accumulate, eventually leading to cell death. If this process were to occur in the brain, for example, it could lead to neurodegeneration. Now, fortunately for us, we don't really have to worry about this, as ERVK integrase isn't typically expressed in healthy individuals. But ERVK integrase has been found to be expressed in certain disease states. My lab has found increased expression of ERVK integrase in the brain and spinal cord of patients with ALS. My research is focused on this ERVK integrase protein and whether or not its expression in previously healthy cells eventually leads to the accumulation of DNA damage, inflammation, and eventually cell death. My goal is to determine if there is a link between ERVK integrase expression and the neurodegeneration we see in ALS. Currently, we do not know what causes ALS, and because of this, there aren't many drugs available for patients. Patients have a progressive loss of muscle function that eventually leads to their death, typically only 2 to 5 years after their diagnosis. If we can determine a link between ERVK integrase expression and the neurodegeneration in ALS, we can look at repurposing already FDA-approved integrase inhibitors as a new drug for patients with ALS who are in desperate need of treatment options. Thank you.